I'm happy and honored to introduce Ben de Haas, who joined us today from Giessen in Germany. He is a group leader there from 2020. Before that, he did his postdoc in the UK. His research spans across different fields, including eye tracking, that I think that we will hear about this today, neuroimaging, individual differences, and I met Ben just a few months ago, actually, in the VSS conference. I was presenting a poster over there and we missed each other, but then he emailed me and we actually already wrote the manuscript. And then he written an email, a very kind email saying, dear Dr. Freud, I'm really sorry, but you have a fundamental error in your data analysis. And then we have a series of interesting meetings. Anyway, the paper is currently written, but we're not going to use it. But I'm very thankful to Ben that thanks to him, we don't have to retract the paper. But it was a great experience to meet Ben and to learn quite a lot from him about the analysis of individual differences. And I thought it could be very interesting for Ben to introduce and present his research here to the CVR members. So thank you again, Ben, for joining us today. And thanks, Laurie Wilcox, if you're there, for organizing the beautiful VSS that allowed Ben to interact with me and to find this issue.
Thank you very, very much for the kind introduction. I have to add to that, that the only reason I recognized this issue in the data was that I did the very same thing myself and very painfully learned. So I actually retracted the paper because of the same, well, conceptually very similar thing. So yes, it's better to catch it early. And the only reason I did was that I had the same thing. That's why I felt compelled to write. So, and Ares was absolutely, I was a bit horrified about writing this email, and I was so glad about your reaction being nice. And I'm very grateful to be here tonight. Well, it's tonight here, it's today at your end. So without further ado, I'm going to talk mostly about eye movements and individual differences. And as you probably all know, when I look at my thumb at arm's length, its width is about maybe 2 degrees visual angle. Now, the human visual field spans almost 200 degrees. But resources in the visual system are distributed very unfairly. On the retina, the density of photoreceptors falls off by 2 orders of magnitude as we go from the fovea to the periphery. This is inherited by visual cortex, where more cortical areas devoted to processing the central few degrees than to the entire rest of the visual field. One consequence of this is that we can only resolve clearly what's at the center of gaze. We constantly need to move our eyes across the scene in front of us. Much like breathing, we can do this consciously, but most of the time, we don't even notice how we do it. Each one of us moves their eyes 2 or 3 times a second. Vision fundamentally is a sequence of eye movements and fixations. This poses a challenge to the visual system. At each moment in time, it needs to decide where to move the fovea next, based on peripheral input. And we still don't really know how this works. There are models, of course, which try to predict which parts of an image will attract our gaze. For a long time, the most prominent ones were variations of the Etienne-Koch algorithm. They all follow the same basic idea. First, extract a set of pixel-wise low-level features like luminance, color, and orientation. Then compute the local contrast for each of these features and combine the resulting feature maps into a single weighted salience map, a general indicator of things standing out in the scene. And that is our prediction of fixation probability. The defining property of these models is that they are completely ignorant towards high-level features. You don't have to know whether there are objects in the image, whether they are a face or a chair. All you care about are low-level image computable features. this was, of course, highly practical and attractive in practical terms, but it also matches the biological textbook model. We know that things like local orientation and contrast can be computed in V1 at the earliest stages of cortical processing. And a prominent model of the visual system poses that after that, we have a division of labor into a ventral stream for what? Am I looking at an object, a face, a piece of text, and a dorsal stream for the where and how. Where is the interesting stuff? Where should I move my eyes next? And the Itti and Kostal models are wholly compatible with this separation. The where can be computed independently of the what, because it only depends on low-level features. The problem is, these models only work for boring images. As soon as you show interesting scenes containing stuff and people, fixations are much better predicted by where in the image is an object. Where is a face, where is a piece of text, and so on and so forth. This study by Xu et al. Used 700 images of complex scenes and delineated and labeled 5,500 objects within these scenes. And they found that this type of semantic information, there is a piece of text, there is a face, carried much more weight for predicting fixations. And these kinds of high-level features have become image computable, of course, with the advent of deep neural networks, which have dominated gaze prediction ever since. So the what and the where do seem to interact after all. And one of the things I'd like to understand is how they do this in the human visual system. Another problem with traditional salience models is that they work with the implicit assumption that two people presented with the same image will fixate the same parts of it. Salience is a function of the image and not the individual observer. Any differences between people are disregarded. These studies typically take the correlation between fixation maps of different observers as an estimate of the noise ceiling. Any remaining differences are deemed random. But are there systematic differences in what you and I look at when we're presented with the same image? I'll try to do a demo now. I don't know whether that works via Zoom. To illustrate this question, I'll play this game, which I hope will work. There will be a subtle change in this image, and I'll ask you to raise your hand or shout, unmute yourself when you think you found it. Okay, Eris saw something. Someone else? Hooray!
I found it.
Okay, so what's the change?
Cheap trick.
Cheap trick.
Yeah, glasses. Glasses.
Oh,
So,
but there's two changes because cheap trick, the text on the drum.
exactly. So the text on the
I saw only one.
and the glasses. So we have two changes, and what we've just witnessed may point to individual variation in gaze behavior. Some of you first looked at the face, and some of you at the text. What you see in this image may not be the same as what I'm seeing, depending on where we fixate. Now, this variation could, of course, be random, like most salience models assume, but by now there are a number of studies that show the opposite. For example, Andrews and Coppola and Bargary et al. Found consistent differences in basic oculomotor traits like saccadic latency, amplitude, and speed. These generalize across tasks to some degree and can even be used for biometric identification. But observers also vary in where they look in a complex scene. Surprisingly, this has a strong genetic component. So I hope I can make this video play. So what you see in this video, hopefully, are the gaze traces of a pair of monozygotic twins, which are remarkably similar to each other and much more so than those of dizygotic twins. So which parts of the scene we fixate and when seems highly heritable, which to me was a big surprise. So given there are systematic differences in where we look, what is the nature of these differences? Could the semantic dimensions that Xu et al. Looked at in their study be used for a general salience model? Well, which they did use for a general salience model. Could this be used to explain the differences between observers as well? To test this, we asked more than 100 observers to look at the same 700 complex scenes, each individual, in any way they wanted. This was literally the instruction to participants, and recorded their gaze. And then we compared patterns of fixations between these observers. So this person was quite interested in the faces in the scene. The green circle shows the first fixation after image onset, so the landing position of the immediate saccade, and the remaining fixations are shown in purple. This observer, on the other hand, seemed much more interested in the hands and in objects being touched. And when we quantify the individual percentage of dwell time spent on faces, we find quite a range across 50 observers. The difference is more than a factor 2. And crucially, these differences are consistent across independent sets of images. If you look at a face more than I do for one set of images, that's an almost perfect predictor for our differences for a second set. Every point in this gray scatter plot is one observer, and you see their individual percentage of dwell time spent on faces for the odd versus the even images in our 700 images. And to the left, you see example fixations by two of these, two extreme observers, the blue observer and the one shown in orange. And these consistent individual differences also hold if we restrict the data to just the first saccade after image onset, the target of this immediate saccade, which we know is under limited voluntary control. So it doesn't seem to be merely a question of someone deliberately looking more at faces, but to point to a more profound difference between your visual system and mine. We find such reliable differences in the tendency to look at faces, text, hands, emotional expressions, objects with implied motion, and food. Not all of them are as consistent as for faces and text, but also clearly not random. And these tendencies are also consistent across time. When we re-invited participants after several weeks, their individual differences replicated. So this seems to reflect a trait rather than a state of the observer. And they go along with differences in perceptual ability. There's a moderate but significant correlation between the tendency to fixate faces and skills in face recognition indicated by the Cambridge Face Memory Test. This relationship with face recognition is corroborated by recent findings of my PhD student Marcel Linker, who collaborated with Maike Ramon at the University of Fribourg. Maike has this database of super recognizers, people who are extremely good at face recognition. In fact, they're so good that Maike is working with the police, and these people are hired to scan CCTV footage for suspects. And Marcel found that super recognizers fixate faces significantly more than controls when looking at scenes freely. And they don't only look more towards faces, but also to objects with implied motion, but less towards text and objects being touched. Interestingly, this push-pull relationship is something we also came across in controls, and which replicated across multiple independent samples. There are systematic correlations between fixation tendencies. Most importantly, the tendency to fixate faces is a negative predictor of fixating text and of objects being touched or hands. And as I mentioned earlier, previous studies have shown that a high proportion of variance in gaze can be explained by genes. But surely the way we look at things is shaped by experience as well. To explore this question, Marcel collaborated with developmental psychologists here in Giessen to study how 5-year-olds look at complex scenes. School in Germany only starts age 6, so this is right before reading onset. And here you see an example overlay with the gaze trace of my daughter, which you see above. And it turns out that the way she looked at this scene is typical for this age group. 5-year-olds spend a lot less time, a much smaller fraction of time of their dwell time looking at text, but they looked at faces even more than the adults do. So we see some of that push-pull mechanism playing out across development and reading acquisition. However, that's not true when we zoom in on the very first fixation after image onset. For this immediate effect, text saliency is rather modest, and I think we don't have a final answer on this yet, because for this study in children, we only used 40 images. You can't make them go for 700. So the resolution we have for the percentage of first fixations is very, very limited, of course. But interestingly, for faces, we still see this effect that even a larger fraction of the immediate fixation compared to the adults is going towards faces. So sometimes it's worth having a closer look. When we throw all fixations in one big bucket, we may confuse different functional subprocesses. We know that the vast majority of fixations is attracted by objects, but not all object fixations are created equal. We think they may serve different functions. Many fixations do come from elsewhere and land on an object to select it for the fovea. In this image, these would be the fixations first landing on the face, the book, and the teddy bear. We call these selection fixations because that's what they do. There's a bunch of objects in the periphery, and we select a subset of those for foveal vision. But both scan path and heat map representations of gaze behavior do not differentiate these fixations from other types of follow-up fixations, like these immediate follow-up fixations on the same type of object. Note there are plenty of these. We call these inspection fixations. So you've already moved the object onto the fovea with the selection fixation, but now you're drilling for further details by slightly shifting the fovea around on that object. And then there are returns, when the eye jumps back to revisit an object after going elsewhere. All of these are typically lumped together, but probably serve different goals. Selection fixations cross the bridge from extrafoveal to foveal vision. Inspection extracts specific details, and returns can refresh our memory or extract further detail from a previously fixated object. And if there is something to this idea, there should be systematic differences between these types of fixations. So Marcel and I started splitting up our data into S, I, and R fixations, and we tested whether across observers, the heat maps for a given image are more similar within one of these classes than between them. So within selection, inspection, or return fixations. And this is indeed the case. Across observers, S, I, and R fixations are much more similar within one class of fixations than between them. And here is another example where this difference is striking. Participants explore quite a few of these objects in the scene, but successive inspection fixations are heavily concentrated on the screen, where smaller details may reveal interesting content, and they mostly return to the face seen in half profile after going elsewhere. And across images, observers are consistently different in how much of their fixation time they devote to each of these classes. Some spend twice as much of their dwell time on selection or inspection as others, and the difference in the tendency for returns are even greater, approaching a factor 4. So the way people inspect a scene is radically different between observers. They vary along a huge spectrum, from drillers, people who spend more than half their fixation time staying where they are, just looking for details on an object they already landed on, to explorers who have no time for that. They spend up to 80% of their time jumping to new objects or hurrying back to one they visited before. The fact that these differences are so consistent across images and observers point to this classification mapping onto something real, I think. It may really point to segregation of functional subprocesses. Another hint of this, capturing something real, is that over viewing time for a given image, there's a steady decline of selection fixations relative to inspection and returns. This matches previous reports of a shift between an ambient mode of a gaze behavior towards a focal mode with longer fixation durations and shorter saccadic amplitudes. But note that this is not the same thing as what we're proposing here. The SIR scheme can uncover steady changes over the duration of a trial, but crucially, it allows us to classify each individual fixation according to its presumed function. And there is no simple one-to-one mapping of fixation duration and saccade length onto SI or R. Fixation durations steeply rise during the first second for all of these fixations, and then they plateau. A little later for inspections and returns, but there clearly is a general trend. The interfixation distance, however, is a bit more nuanced. Unsurprisingly, inspection fixations, which by definition are on the same object, are always nearby. But selections start with huge jumps across the image, to then, after about a second, drop to about half that distance, while returns climb up to that plateau from a much lower level and more slowly. In other words, when we return to objects early in the trial, we only do this if they're nearby. But later on, we can be compelled to cross larger differences to go back. And this distinction that we propose between these SI and R fixations matters when we try to understand which parts of an image attract fixations. When we fit a salience model separately to each type of fixation, we see that some of the predictor weights vary wildly between them. Whether or not a face has an emotional expression makes no difference for selection fixations, whether we fixate the face at all, but emotional expressions strongly compel us to drill deeper on the face with inspection fixations and to come back with returns. Similarly, and not surprisingly, the salience of text objects is mainly due to inspection, because this is the way reading works. Once we are fixating a piece of text, we're very likely to make follow-up fixations on the same piece of the image, of the same text object. And finally, a cool technical aspect of this is that we can approximate SI and R fixation maps pretty well without metadata on the location of objects in the image. Remember, we only could do all of this because we're using these stimuli that come with metadata telling us which pixels in the image belong to the same object. But what if you don't have that? If you use an image set without object masks? We found that the SIR classification of fixations can be approximated very well with a simple spatial heuristic, which is completely agnostic towards image content. We simply define a Euclidean distance threshold to guess whether a given pair of fixations landed on the same object or not. We found that 10% of the overall image width is a very good proxy, which leads to surprisingly similar results to the analyses using object masks and labels. So as long as you know the order and locations of fixations, that's all you need to readily apply this classification. And we hope this will be useful, not just for our own research, and we will publish the data and the code along with the paper once we get someone to accept it. So if this sounds interesting to you, stay tuned. Another tool that may be helpful for others, hopefully, is a short test of individual gaze biases. When Marcel collaborated with Maike to test super recognizers, these participants saw the 700 scene images introduced by Xu and colleagues. This yields very good and rich data, but it's a long experiment, about 45 minutes in total. If we want to go beyond a typical psych student sample and test large samples or special populations, we need something more efficient, something shorter. So Marcel set out to find and validate a subset of images for a quick test. And it turned out that, at least for the most important gaze biases towards faces and text, you can get decent estimates with just 40 images. Each one is seen for 3 seconds. So the test literally takes 2 minutes net viewing time. This is published, including materials, so if you're interested, please go ahead and use it. For us, this already opened up a few interesting avenues. One is the kids study I mentioned earlier, but Marcel is also collaborating with clinicians from the University of Marburg, which is nearby, using his short test in children with autism spectrum disorders, ADHD, and schizophrenia. There are reports that gaze behavior may be diagnostic for these types of conditions, but to decide whether gaze can serve as a diagnostic marker, we need to do better than a few dozen controls. So we're also collaborating with Mathematicum, a local science museum, which at least pre-COVID had 100,000 visitors per year. And Marcel developed this interactive exhibit in which you can self-administer our short tests. And once we're out of this pandemic and they can open again fully, we hope to gather about 10,000 data sets this way within a year, which would go a long way in the direction of a benchmark sample to compare clinical groups to. But are these dimensions we identified and we're focusing on really targeting the dimensions of individual gaze? How much of the variance in object fixations can they explain? To answer this question, we need to go back and forget about the semantic labels like faces or text. We note the dwell time for each individual object among the thousands of objects in the image. So for a given observer, we get a long, sparsely populated vector because most of these objects were not fixated at all by this specific observer. And I think here I forgot to update a slide. This is showing my previous host from Bailan. It should show Eris now. Now for a pair of observers, like Eris and me, we can compute the Euclidean distance between our vectors. This is a simple metric and a completely agnostic metric of how different we were in distributing our fixations across objects in the image. It's independent of any sort of label. And if we do this for 100 observers, we get an observer similarity matrix, showing the distances between all pairs of observers. It shows us who is more and less similar to whom. The first question is, how much of that is consistent? If Erez and I are particularly similar in our fixations for one set of images, will we also be among the more similar observers for a second set of images? And the answer is yes. The difference between observers for odd and even images, the inter-observer differences, correlate with 0.9. If you and I are particularly different for one set of images, we'll also be among the very different pairs for the other set of images. So that's our benchmark. 80% of the differences in object fixation are potentially explainable by dimensions we can come up with. How much of that do we actually capture with our handful of dimensions? We tested this by fitting a linear model. We tried to predict the overall differences in object fixations as the weighted sum of the differences in the tendency to look at faces, text, and so on. Because some of these predictors are correlated with each other, we use ridge regression to nudge the model towards a sparse, efficient solution. And we make sure to cross-validate. So we learn the weights for these predictions in one set of images and then evaluate the model predictions in the remaining images. And this way, we can capture 27% of the explainable differences in object fixations with a handful of dimensions. The bulk of this is down to differences in fixating faces and text. So these dimensions do seem very important for our differences, but at the same time, we're still far from capturing everything that's going on. We're only explaining less than a third or about a quarter of what we could potentially explain. So what else may be going on? My PhD student Max Broda is collecting online ratings on the perceived valence, arousal, attractiveness, age, gender, and several other features of all the people in these images. Maybe we don't just differ in our tendencies to look at people, but more specifically in our tendency to fixate happy or sad or attractive faces. And Max and mainly our poor student research assistants also undertook the heroic effort to expand the pixel map annotations for all depicted humans. Across images, there are more than 1,000 people, and we now have hand-drawn delineations and labels for every head, eye, mouth, hand, arm, leg, torso, and the inner face region. So that's an additional 6,000 pixel masks, which is more than the total of the original dataset. The most amazing thing about this is that most of our research assistants are still willing to work with us and don't seem to hate me yet. And when we publish this, we'll make all materials available so the community can also benefit from this enhanced version of the metadata Fisk-Zoo dataset. And using these new annotations, Max found that observers show very consistent and large differences in their tendency to fixate these features, most prominently eyes and mouths. And he also confirmed a push-pull mechanism between fixations towards hands and faces, which we assumed because we had this for touched objects earlier. And something similar is going on when we zoom in on face fixations. If looking at the face, you tend to fixate the eyes more, you fixate the mouth less. This replicates a finding by Matthew Peterson, showing that the preferred height of fixations towards a face is individually different. Face-directed saccades of some people tend to land in the eye region, and those of others are shifted downwards, sometimes as far as the mouth. Importantly, Matthew Peterson has shown that this landing point is individually optimal. Observers perform best at face identification when they are free to saccade to their preferred landing position. What's more, Peterson later found in a painstaking mobile eye tracking study that when people spontaneously fixate in the lab on a screen is an almost perfect predictor of gaze behavior in the wild. Eye lookers consistently look higher up in faces they encounter in real life, while mouth lookers consistently fixate further down. But hang on, how can this possibly be? Why is it seemingly better for some of us to fixate the eye region and for some of us to fixate further down? Why are some of us eye lookers and others nose or mouth lookers? There are two main hypotheses. The first is that face recognition uses an internal face template, which is stored in retinotopic coordinates and shifted between observers. We then look at faces in a way that tries to bring the face on our retina and register with this internal template. There is some evidence that neural tuning for different facial features is distributed unevenly across the visual field, and that observers show idiosyncratic visual field biases in face perception. If we have individually shifted face templates, we may either be born with them or learn them over time. For instance, if you find eye contact unpleasant, you may start looking further down in faces for that reason. But over time, this will shift your internal face template in that direction, and it becomes perceptually ideal for you to fixate this way. We may start looking at faces in different ways for all sorts of reasons. Where we look in a face, and specifically whether we look at the eyes or not, has been the focus of cheesy attempts at romance, poetry, and never-ending stream of pop songs, cultural conventions, and research into social cognition and clinical tendencies and conditions such as social anxiety or autism. An early decline in the tendency to fixate the eyes has even been proposed as a biomarker for autism. So we may start looking at faces for reasons differently at faces for reasons that are not perceptual in origin, but over time this could shape our face template and ultimately face perception. However, there is an alternative hypothesis. The difference between eye and mouth lookers may not be face specific at all. We know that beyond the general drop-off of visual fidelity in the periphery, there are idiosyncratic visual field biases. One observer may have a slightly higher cortical resolution in the upper fovea compared to the lower, whereas it may be the other way around for the next person. Such low-level domain general biases, of course, would predict similar differences in gaze behavior. Placing faces in such inhomogeneous visual fields someone with higher fidelity in the upper visual field would do well to fixate lower in the face and vice versa. So both hypotheses predict the differences we see in face looking. However, they do differ in their prediction regarding the relationship between face fixations and the way we look at stuff. If the difference between eye and mouth lookers is down to shifted face templates, the way we look at a can of soda should be unaffected by this. It should be independent. If, however, the difference between eye and mouth lookers goes back to domain general differences in low-level processing, they should generalize to all types of stimuli, not just faces. The way you look at a can of soda should be a good predictor of whether you're an eye or mouth looker. So here's a straightforward experiment. In fact, we were able to reuse data from an experiment we recently published already, which was quite fortunate given COVID and that we stopped all eye tracking for a long while. So in this experiment, 101 observers freely viewed these 700 scenes, like the one you're seeing here from this data set from the OC. They had their head in a chin and forehead rest, so the image vertical was aligned with their vertical meridian. And we eye tracked them with an iLink 1000 plus, defining fixations using standard criteria. Each image was shown for 3 seconds in blocks of 100 images, which were followed by recalibration. And given we now have all these masks for all the inner face regions, mouths and eyes, we can extract all fixations landing on the inner face region and determine their height relative to the vertical distance between eyes and mouths. A fixation landing exactly at the height of the eye region would be coded 1. A fixation bang in the middle, 0.5, and this fixation below the mouth as minus 0.2. Similarly, we coded all object fixations according to their height between the vertical extreme points of the objects they landed on. And once we do this, we can pull all the fixations of a given observer across all images and calculate the average height of fixation landing on objects and the average height of fixations in the inner face region relative to eyes and mouths. So here we see the face fixations of 2 extreme observers. The red one at the top clearly prefers the eye region, while the blue one at the bottom tends to land in the mouth region. The green scatter points show the average relative height for faces and objects for all fixation, for the first fixations of all the 101 observers. And you can see that there is a clear consistency in the individual biases between faces and objects. And this shows in objects as diverse as spoons, mugs, or mobile phones. Our blue example observer clearly likes to fixate lower down in these, whereas the red observer prefers higher landing points. Note that these images were shown at a size of 29 by 22 degrees of visual angle, so these differences are large compared to the accuracy of the eye tracker and calibration procedures. Preliminary data also show that these differences are stable across sessions. Sorry, the slides are running on their own. Across sessions that were separated by several weeks, which we think renders a calibration artifact really unlikely. The finding that fixation behavior towards faces is predicted by fixations towards stuff is in line with the hypothesis that both are caused by domain general visual field biases. However, our data don't tell us which ones they are. So that's what we plan to do next. We want to test a number of candidates, including visual field bias in acuity, contrast, and crowding sensitivity. Our findings also don't tell us how to make sense of the fact that eye avoidance correlates with aspects of social cognition. In our sample, individual differences in gaze behavior towards things and faces share a little more than half their variance. So there may be room for an independent social factor, which probably would become more salient in situations outside the lab. And even though it may seem unlikely, we currently cannot rule out that low-level visual field biases have a relation to social cognition. So, as always, further research is needed. And with that, I'd like to thank you again for your attention our funders and collaborators, but most of all, my wonderful team that managed to do all of this in these crazy 18 months of a pandemic and made setting up a lab fun. The PhD students, Marcel Linker and Max Broder, whose work I presented today, Petra Borowska and Diana Kolenda, and our Click Warriors student RAs, Diana Weisleder, Tamara Alzheimer, and Liebig.